turbo engine running.
Absolute genius.
Get this.
Welcome.
This is the show where we bring you
Science.
What that essentially means is
discoveries,
Advances.
Questions. Research.
technology. Unbelievable.
Without further ado,
This is The Naked Scientists. Hello, welcome to The Naked Scientists. This is the show that brings you the biggest breakthroughs and talks to the major movers and shakers in the worlds of science, technology and medicine with me, Chris Smith. Coming up, evidence of a carbon cycle on Mars has been unearthed by the Curiosity rover. So what does it mean for the red planet's past habitability? Also, the cannabis-based painkiller as powerful as an opioid but without any of the side effects. And could fashion sense and a primitive sunscreen have been the deciding factor, factor 50 in fact, which allowed us humans to outlast the Neanderthals?
From Cambridge University's Institute of Continuing Education, this is The Naked Scientists.
First this week to Mars, where the team behind the Curiosity rover, which is trundling around a site called Gale Crater and using a range of instruments to analyse surface samples, have announced the detection of significant deposits of iron carbonate minerals. The discovery supports our view that the young Mars had a dense carbon dioxide atmosphere that made it, at least periodically, a warm, wet place with, therefore, many of the essential ingredients for life. From the University of Calgary, here's Ben Totolo.
the one thing that has really driven research into understanding Martian habitability over geologic time is that it's further from the sun. It gets less of the sun's rays onto its surface, and therefore it's even harder to heat than Earth is. And so it must have had a lot of carbon dioxide in its atmosphere in order for all of these signs of liquid water that we see to be there. And what we found is that for the first time in Gale Crater and really anywhere on Mars, that there's abundant carbonate minerals in these strata, so in these sediments. And what that mineral allows us to think about is what the atmosphere was like at the time that these sediments were deposited.
How do you put a timeline on this? So when you find a particular stratum and you find a mineral in it, how do you date that?
On Mars, that's a very challenging thing. So Mars, we believe it was habitable up until about 3.5 or 3 billion years ago. So that's when we believe most of the evidence of liquid water was deposited. And that's largely or almost exclusively based on counting craters on the surface of Mars. So it's a fairly inaccurate or at least wide uncertainty type of measurement. And the thing that we know is that the sediments that we're investigating now must be younger than the sediments below them, basically.
And presumably, when you know something about what was going on in the water, that must tell you something about what was going on in the atmosphere, because the two are so strongly linked.
Yeah, 100%. So we believe that Gale Crater was once a lake, and all of the sediments that we're looking at were deposited in a lake or a lake that was transitioning to sort of more of an Aeolian type deserty environment. Based on that, we know that there must have been water there in the rocks that had interacted with the atmosphere. And the thing that we propose in this paper is that something must have changed in the atmosphere, not the carbon dioxide concentration, because we believe that had to be high already in order for the planet to be hosting liquid water, but rather something about, for example, the sulfur concentrations that enabled the waters to actually begin precipitating carbonates. The other thing that we have hypothesized from the geologic evidence is that this is a lake that was drying, so its ingredients were becoming much more concentrated. So it became more easy to precipitate minerals like carbonates and then also the other minerals that occur here, which are magnesium sulfate salts, so super highly water-soluble salts.
So returning to your opening question, which is trying to investigate the timeline for when Mars might have been habitable, is that at all compatible, that timeline, with the possible emergence of life processes, or did Mars become uninhabitable too young?
When I started working on the mission, that I had a very different opinion about how Mars habitability, how long it persisted, and then also how constant it was. I think the emerging picture from the community is that the habitability was intermittent, right? So it might have been warm and wet for a while and then cool and dry again in alternating cycles until this permanent drying and cooling that happened around the time that these samples were precipitated. So overlaid on that, the timescale of how long it takes to do the biogeochemical reactions that lead to the building blocks of life. And very interestingly, some of that research shows that, maybe even a handful of weeks is too long for some of those reactions because of things like being bombarded with ultraviolet radiation on the early planetary surface. So the idea that's emerging for me is that even though Mars wasn't persistently habitable over its entire history, it would have had long enough duration of liquid water, long enough duration of warm and wet conditions for the origins of life to have occurred. The question is whether that ever happened or not.
And are we in a position to detect them?
Yeah, that's the other question that we've been thinking a lot about lately. So we've been working with others here at the University of Calgary, thinking about how actually these organic molecules would be preserved over geologic time. And that seems to be quite a challenge. I think over the last few weeks, another finding from the Curiosity rover about long chain organic molecules was announced. So there's some evidence that the things that go into making the first biomolecules in cells are even present now in Gale Crater. But again, the problem is that most of these sediments were deposited more than 3.5 billion years ago. And they've been, ever since then, getting bombarded by the ravages of geology and the ultraviolet radiation. And so it would be nice to predict that someday we're going to see the smoking gun biofossil that really indicates that the first cells were there. But in the end, we're often basing what we can see on inference instead of actual detection of the real thing.
In the past, though, we have detected carbonates of the sort that you found on Mars. They have been found, although not in great quantities. So why is your discovery here? Why is it a big step forward?
So the context for the other carbonate discoveries to date have been that most of them weren't pure iron carbonate. So what we see here in Gale Crater is effectively FeCO3. And that's predicted based on our understanding of evaporating Mars waters. That's what we should see. So what we believe is that this is the first time we're truly seeing what are sedimentary carbonates. On Earth, sedimentary carbonates are often limestones, calcium carbonate, right, like the White Cliffs of Dover. Here on Mars, because the crust is much more iron-rich, we predict iron carbonate. And those detections to date have often not seemed to be sedimentary carbonates. They seem to have been replacement of minerals that might have occurred at high temperatures through hydrothermal processes, and not necessarily through the sedimentary deposition processes that we predict would characterize a carbon cycle on ancient Mars. And so the big discovery here is that we were able to use this amazing instrument on the Curiosity rover, the ChemIn instrument, to detect not only the abundance of carbonates of 10% or higher, and that's remarkable, but also the chemistry of carbonates in terms of not having ever seen anything like that before.
I suppose it also validates your model, doesn't it? In the sense it gives you confidence when you make predictions or also decide where next to look and what to look for. You can say, well, we've got some degree of confidence in what we think was going on and when, based on now these other detections.
We were completely surprised that there was this much iron carbonate there. In principle, it's present in enough volumetric abundance that we should have been able to see it from spectroscopic studies of the Martian surface. But we didn't. And we went back even after this discovery to see if it could have been detected and we just missed it. It's not apparently there. So the interesting thing here is that we have this in situ or ground-based measurement on Mars that shows something different than what we could have seen from orbit. And then based on that, we can look at other similar deposits around the planet that contain this magnesium sulfate mineral that is probably masking the presence of iron carbonate and say that actually probably all over the planet, this iron carbonate is there. We just haven't seen it because we haven't had rovers everywhere.
So what else are we missing?
Yeah, that is, to me, the biggest question is what can't we see in the orbital data that is probably there on the ground? And that would actually help us to rewrite the history of habitability on Mars.
Fascinating insights into Mars's past. Ben Totolo there at the University of Calgary. That work just came out in the journal Science. Treatment for severe pain still relies very heavily on opioids, in other words, morphine-like agents. These are very effective in the short term, but they're also highly addictive and potentially deadly in overdose or if otherwise misused. Led by the fact that many people find relief with cannabis, US researchers have now come up with a compound that mimics one of the molecules found in cannabis. But the clever twist is that by making their new molecule more water-loving, it can't get into the brain to produce any mood-altering or addiction effects. Instead, it works purely by activating cannabinoid receptors around our peripheral tissues. The effect doesn't appear to wear off with repeated use, And although they've tested it only on mice so far, it does also seem to be incredibly potent. Here's Washington University's Richard Slavicki and first Sushruta Majumdar.
The questions we were trying to answer is, can we separate the psychoactivity or the mild altering properties of cannabinoids, but retain its pain-relieving properties to develop a non-opioid analgesic or a pain reliever?
Because when we talk about cannabis, that's actually a cocktail of compounds, different chemicals, isn't it? And so are you saying then that the ones that give you the mind-altering, mind-bending effects are not the same as the ones that cause pain-killing effects?
Yes. So what we are using here is a single entity. We're not using phytocannabinoids like are present in medical marijuana, for example, but we are tweaking the chemical structure in a way so you can separate the mind-altering properties from the pain-relieving properties.
Rich, how are you actually doing that? How do you dissect apart those two effects?
One way we can do this is target cannabinoid receptors that are located outside of the brain and the central nervous system, targeting tissues that are feeding in those pain signals into the central nervous system.
So in other words, the pain-killing effect is not necessarily achieved in the brain. It can be achieved outside the brain, but the mind-bending effects are achieved inside the brain. And if you go for the two different targets, you can isolate those two effects.
Absolutely. Yeah, that is the point. So what we did was we just make subtle chemical changes into this molecule and make sure that it only targets the periphery. There's something called the blood-brain barrier that the body naturally has to prevent drugs and harmful chemicals from entering the brain. And the blood-brain barrier needs oily compounds or greasy compounds. So what we did was with the synthetic cannabinoid we made it more water-soluble or less oil-like, as a result of which it will not enter the brain and only target the peripheral nervous system or tissues outside of the brain. And you're able to achieve the pain-relieving properties while you're able to separate the mind-altering properties.
How did you test it, Rich, to prove this was going to work?
So in order to evaluate this for just pain relieving properties, we use animal models of pain. And so we use different models of migraine, neuropathic, and inflammatory pain. Inflammatory pain is just like an acute injury due to like a burn or like an infection. Neuropathic pain is more of like a chronic pain, and so something due to like nerve damage. We then screen these molecules for not only their pain relieving properties, but also their potential for side effects in rodents. And so we can evaluate those, which might equate to the mind altering effects of cannabis, which is something, again, we wanted to avoid.
And do you get a suppression of pain responses in these animals, which would suggest that this is working, but without producing the psychoactive effects?
We find efficacy across all three animal models of pain. And importantly, we didn't see any analgesic tolerance. That is, with repeated dosing, the compounds still retained pain relieving effects, which is really important because with opioids and even cannabis, we see this tolerance like effect. And more importantly, too, we found a great separation between the pain relieving properties and the centrally mediated effects, which indicates that this compound has a really wide therapeutic window, which is really important for kind of a first in class analgesic.
And Sush, one of the things about the opioids that makes them very attractive is they are incredibly potent. We can achieve really powerful control of pain with them, which under certain circumstances we really need. So are these drugs any good at rivaling the opioids for that?
That was one of the surprises of this project, that these turned out to be extremely, extremely potent. And I've been working in the opiate field trying to develop drugs on that side of things also. And you're absolutely right. The potency has always been an issue with the other targets, but these were as potent, if not more potent than morphine, which is the choice for treating pain, or even fentanyl for that matter. And the beauty of this system is that unlike the opioids, they do not produce respiratory paralysis or loss of breathing and the non-addictive properties because we are targeting the receptors outside of the brain. Sedation is a huge issue. Stoning is a big issue with the cannabinoids or psychoactivity, mind altering. There was a 100-fold separation between the pain relieving properties and the adverse side effects like sedation with this class of molecules.
Rich, have you unpicked how exactly you're doing this? Do you know why those pain nerve fibres have those receptors on them? Why should those pain systems have the ability to respond to this class of compounds at all?
The body has its own cannabinoid-like system called the endocannabinoid system. So we actually synthesize molecules that bind to these receptors. The body has these receptors throughout itself, and it has various different roles and different physiology. Stress-induced analgesia is a big one. There's a lot of like pain inhibition that goes on with these endocannabinoids endogenously.
So this is presumably quite a big step forward then, because you've got something which appears to give opioids a run for their money in terms of how good it is at painkilling, but doesn't seem to have any of the negative baggage. The thing is, it's one thing to have a molecule in a test tube, but a test tube into a needle in a patient's arm, or even a pill going down their throat, that's a big hurdle yet, isn't it? So you think it's surmountable, though? It looks promising.
Right. I think it's very promising. I think one of the challenges we have had in the pain field has been to identify targets which, as you pointed out initially, have the potency of the opioids, but with the non-addictive potential or the respiratory depression potential. So we have identified a target. The target has been de-risked. And the next step is to modify this molecule or a second generation molecule and convert it into a pill. This is definitely doable.
Hugely promising. Thanks very much to Richard Slavicki and Sushruta Majumdar there. They've just published that work in the journal Nature.
The Naked Scientists podcast is produced in association with Spitfire, cost-effective voice, internet and IP engineering services for UK businesses. Find out how Spitfire can empower your company at spitfire.co.uk.
Music in the programme is sponsored by Epidemic Sound. Perfect music for audio and video productions.
This is the Naked Scientists podcast with me, Chris Smith. Still to come were more primitive clothing and a lack of sunscreen coupled with a reversal of Earth's magnetic field, what may have done for the Neanderthals. Before that, though, a new blood test called Pac-Man to help detect pancreatic cancer earlier has been engineered by researchers in America. Pancreatic cancer is notoriously hard to spot early. It usually presents very late and with a correspondingly grim prognosis. It can also be hard to distinguish in the initial stages from some other forms of pancreatic disease, muddying the diagnostic waters. The new test looks for pancreatic protease enzymes that have spilled over into the bloodstream where they should never normally be. It can correctly distinguish patients with pancreatic cancer from non-cancerous problems 98% of the time, and used alongside existing cancer diagnostic markers, it can spot early-stage pancreatic cancer with, they say, 85% accuracy. It's the brainchild of Jose Montoya and Jared Fisher from Oregon Health and Science University.
So there's currently no good screening method for pancreatic cancer. It's really typically only detected accidentally from an unrelated medical condition, and also the early warning signs are very vague, such as just you have pain in your abdomen, you have yellowing of the skin, fatigue, recent onset diabetes, weight loss. And these kind of conditions are very common for many other types of diseases outside of pancreatic cancer.
So what did you do, Jose, to try to come up with a better way to find this and find this early?
It was clear to us that priority was early detection, meaning that we needed to be able to detect that transition from healthy tissue to malignant or cancer disease. And that meant that we needed to find something that was present in early stages and later stages as well. We decided in a type of protein that is called a proteases, which, if you just allow me this analogy, they sort of work like a really well-regulated pair of scissors that the body uses. The body uses proteases to chop proteins when they are not needed. And one of the most common ones is in your stomach when you ingest food. These proteases basically digest the food and cut the protein so then the gut can absorb those nutrients.
And the pancreas makes one of these proteases, and you're saying, well, let's use that because if it's in the blood, it shouldn't be. So therefore, it might be a marker of something being amiss with the pancreas.
That is exactly what it is. So we actually found that even the cancer uses these proteases in order to grow. So then we can utilize these markers, these proteins that are present in circulation, and look at their activity in order to know how much is the cancer progressing or at what stage is at.
So Jared, when you do this, how much better is it than what we have at the moment, which is not very good by the sound of it? When can you pick up cancer and how early?
So how we addressed that initially was what's called retrospective, or these are samples that are already collected and are frozen and that are found at different stages, which are different levels of the disease, of the pancreatic cancer. So stage 1 is considered a disease that's very local and confined just to the pancreas and can go all the way up to what we call stage 4, where it is spread throughout the entire body. And we found that even when the disease is just localized to the pancreas, we get very, very strong detection. And then the other aspect is how many of the patient's samples that have cancer do you call correctly? So that's called sensitivity. 73 to 85% of the time, we can correctly call samples that have cancer. We say that we are highly probable, highly likely that they have cancer.
The key thing is, though, it's got to be very early. So therefore, how sensitive is it and how likely is it to make a difference to those people who could use this as a screening test and find they have very early pancreatic cancer that would then be amenable to curative or potentially curative treatment?
So in the general population, your chance of getting pancreatic cancer is well less than 1%. But in a subset of patients that have pancreatic damage, or even patients with diabetes, their chance of getting pancreatic cancer is elevated to 2%, upwards of 20%. And what we showed is that we never call those patients with these pancreatic inflammation, pancreatic disease that's non-cancer, we never call them with cancer.
Basically, you've got a really good rule-out test here. So if you see something going on and you apply your test and it is not positive on your test, it's almost certainly not cancer, and therefore you can reassure the patient, but it also means that you don't then go hunting for something they haven't got. That's the critical thing, isn't it?
Yeah, that's where, especially for early tests, there's two ways of thinking about it. Do you want to make sure you pick up all the cancers, which is one way of thinking about it, but you also then you never want to call a healthy person to say that they have cancer. So we're very good at that aspect.
Jose, how can you take this forward? You've got a good rule-out test. It looks like you can call a spade and say this is not cancer, but you're slightly less good at picking up cancer is there, but it's still very, very good if it does pick it up early. But how can you now apply this? Is the next step to do a really big proper clinical trial rather than a retrospective analysis?
We're exploring 4 different avenues right now to try to grow this study. One of them is to make a bigger study similar to the one that we're doing, but we're actually going to use other hospitals, so patients from other hospitals, and not just other hospitals, but also other countries, since part of what we want to do is be able to use this not just here where we are, but everywhere in the world. A second one is reiterating on what Jared was mentioning, is using this high-risk patient cohort. We're hoping to start a clinical trial, a proper clinical trial, like you were mentioning, where these high-risk patients that have inflammation in the pancreas or other diseases, and hopefully we're able to pick up that some of these patients are transitioning from a non-pancreatic cancer to a pancreatic cancer state. We also have the idea of expanding to other cancers. Foundationally and fundamentally, if you look at the biology of these proteins that we call proteases, they are also present in many other cancers. The other cancers use them for the same purpose or slightly different purposes. And I think we can exploit that difference to be able to, again, create early detection tests for other cancers. We are also going to keep improving the technology. The Pac-Man test that we developed, we need to keep working on it so we can have the best foundation for all of these new findings that are coming.
Jose Montoya and Jared Fisher there. So why is their test called Pac-Man? Well, they tell me protease activity was part of it, and a penchant for 1980s computer games that the pair of them shared did the rest. They published that work in Science Translational Medicine. Until 40,000 years ago or so, there were 2 groups of human ancestors alive on Earth. Us, anatomically modern Homo sapiens, and our close relatives, the Neanderthals. They didn't just overlap in time either. There's evidence that the 2 inhabited some of the same territories and even interbred. We've all got a small number of Neanderthal genes in us. But suddenly, after hundreds of thousands of years, Neanderthals abruptly disappeared. A range of theories have been advanced to account for where they went. These range from competition from our forebears through to absorption into a larger, more rapidly expanding, anatomically modern human population. But now, scientists have another tantalising hypothesis. 41,000 years ago, the Earth's magnetic field flipped, or at least looked like it was about to. Known as the Le Champ excursion, this temporarily dialed down the magnetic force field that defends us from space radiation. This would have thinned out the ozone layer, so UV levels would have surged. With their more advanced clothing and a familiarity with iron-based makeup, perhaps our anatomically modern human ancestors were better equipped to deal with it, suggests Raven Garvey, an anthropologist at the University of Michigan.
Currently, Earth's magnetic field is what's referred to as a dipole. There is a North Pole and a South Pole, but these poles reverse periodically on roughly a timescale of every 200 to 300,000 years. But sometimes Earth's poles will wander a bit and become unstable. This particular group of scientists were interested in a phenomenon about 41,000 years ago, where the pole came near to reversal, but didn't quite. So the dipolar structure of the magnetic field was quite disrupted. And this likely had near-Earth implications. So things that would have been felt by all sorts of life on Earth, which is where I became involved. As an archaeologist, I study such things as humans' interactions with their environments.
What was the sort of population like 41,000 years ago? Who was around and what were they doing and where?
Just to give a little bit of context, archaeologically, it can be quite difficult to reconstruct demographics, to know exactly how many people, or even approximately how many people, were in a place at a particular time. We believe that populations were quite low. The area where this Le Champ phenomenon, this excursion of the magnetic poles, would have been quite strong was Eurasia. And so living in Eurasia at the time were two species of hominins, the Neanderthals and Homo sapiens, which is us.
And were they overlapping? Were they interacting? What do we know about what they were doing at that time?
Neanderthals arrived first, so they probably developed in Eurasia sometime before 400,000 years ago. Evidence currently suggests that Homo sapiens, anatomically modern humans, arrived in Eurasia probably about 57 or so thousand years ago. And just about the time of this Le Champ excursion, so round about 45,000, 43,000 years ago, anatomically modern humans spread westward quite rapidly, and there is evidence that they were interacting with Neanderthals. Genetic evidence shows that many living populations of humans, Homo sapiens, us, have between 1 and 4% Neanderthal DNA. So there was certainly some amount of interaction going on when these two populations were living side by side in Eurasia.
But critically, that's also the time point when we know that Neanderthals began to disappear, isn't it? Because if you look much more recently than that, they've gone quite quickly. So are these results suggesting that this phenomenon to do with the reversal of the magnetic field might be linked to that happening?
That's precisely what we're interested in exploring. So, yes, in fact, Neanderthals are no longer around in this area by 40,000 years ago, which is right about the tail end of this geomagnetic phenomenon. So, one of the things we've wondered is what about that phenomenon changed what people in this place might have been experiencing at the time? And one thing that may have occurred is that the dipolar structure of our current magnetic fields creates something like a force field around our Earth, which ordinarily shields us from an awful lot of bombardment of particles from space, including ultraviolet radiation, which you may have heard in other contexts is what causes things like sunburn and skin cancer. And so when the dipolar structure sort of broke up, the magnetic field lines were not creating that shield nearly as effectively. So more of this ultraviolet radiation was making its way to Earth. And so one of the things we wonder is, are there differences in the cultures practiced by Neanderthals versus Homo sapiens that might be significant in this regard? Archaeological sites that we associate with Neanderthals have lots and lots of tools, and some of those tools were probably used for the preparation of hides, probably for things like cloaks or loincloths, maybe some footwear to protect their bodies, because this was also a very cold time in Europe. Sites that we associate with Homo sapiens, conversely, not only have similar kinds of tools that were for dispatching animals and removing their hides and preparing those hides for other purposes, but we also find needles and awls, things that we associate with stitching, with sewing. And so one interpretation of this difference is that Homo sapiens were making tailored clothing, clothing fitted to the limbs like shirts and pants, rather than simple draped clothing that we tend to associate with those Neanderthal sites. And this may have conferred the advantage not only of keeping people warm in what is a cold climate, but if the UV radiation is, as we suspect it might have been, stronger during the Lachamp excursion, this would have provided a secondary benefit, that of protecting exposed skin from harmful radiation. In addition to the tailored clothing, in sites that we associate with Homo sapiens, there is considerable amount of a substance referred to as ochre, which is iron oxide, that in some modern populations is used as a sunscreen, and there have been experimental studies to show that it is an effective sunscreen. So the fact that the amount of this substance in sites associated with Homo sapiens goes up during this space weather event, the fact that it has been observed as an effective sunscreen in more recent times is again suggestive that people may have been applying this to the skin as, say, another form of sun protection.
Is there any evidence that they were doing that, as in artwork or other documentation where you can see people were clearly applying this at that time, which would add kind of weight to the idea that people were self-medicating almost in that way?
Sadly, no. I wish we had such evidence, but the depictions at this time, depictions in rock art, are primarily of animals or abstract designs, and that I know of, there are no representations of hominins, human-like forms applying a substance to the skin.
And if that won't convince you to take better care of your skin, I don't know what will. Thanks very much to Raven Garvey from the University of Michigan. She's just published that study in Science Advances. And now it's time for Question of the Week. And James Titko is doing his best Dr. Dolittle for us this week as he tries to speak the language of the animals for this query from Kieran.
Although animals, especially dogs, cats, and some birds like parrots, can speak or understand the English language, how are they still able to follow human instructions so effectively? Thank you, Naked Scientists. Bye.
Thanks, Kiran. Human languages contain words with very specific meanings incorporated into tight grammatical structures. Our friends from around the animal kingdom, some of them can understand vocal calls, including our own, but they're also relying on a host of other cues. Here to help me answer your question is Nikki Clayton, Professor of Comparative Cognition at the University of Cambridge.
When a human being gives a vocal command, there's a lot of information available. Abercrombie once said, "We speak with our vocal organ, but we converse with our whole body." When we give a signal, it's not just the acoustics of the word, hello, 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 the different sounds, but also the signal would include the body posture. It may be opening, it may be closed, they may be smiling, they may be looking somewhat afraid. And the gestures, the hands might be open or closed. The eye gaze cues, are they looking at the animal, are they looking away? Whether you're scared or embracing, whether you're ebullient, all those kinds of things will be conveyed by the nature of the way in which we speak, rather than just the words that we utter.
But to what extent do animals understand our vocal commands, rather than just associate them with certain outcomes?
Well, I think there's also a difference between whether you speak the word yourself or whether you understand it. For those of you who have little children, you'll also know that little children understand things earlier than they can form eloquent sentences of things. We do know that a number of animals, such as dogs, are very good at understanding human commands. Sit, for example, or come here. Perhaps what some of the listeners don't know is that in my lab, in our work with members of the crow family, that includes the rooks, the jays, the ravens, the magpies, well, some of those are also able to understand human commands. speak, wait, come here, responding directly to the sound of the word itself, irrespective of who has given that command and irrespective of whether they can see the facial movements of the person giving the command.
So, Kieran, rather than always knowing the meaning of the words, animals may be using other signals like tone, body language, and gestures to interpret what we mean. But many animals do seem to be able to understand the commands we use, and to what extent is an active field of study. Thanks so much for sending that one in. Next time, we'll be answering this one from another of our listeners.
Hi, it's Caroline. I've been listening to your podcast on measles. I wondered whether the ability of the measles virus to wipe out or reset the immune system has ever been studied as a potential therapy to help people with autoimmune disease. Could the immune system be reset and reprogrammed so the body no longer attacks itself?
And if you think you know the answer, or if you would like to send in a question of your own, it's chris@thenakedscientist.com. You can also find out more and follow the discussion on our forum at nakedscientist.com/forum. Thank you very much to those of you who continue to support the programme with donations. You can do that at nakedscientist.com/donate. We're really, truly grateful to those of you, including those of you who give us a donation every single month to support what we do here at the programme. So if you like what we do and you enjoy listening to this programme, do please consider heading over to nakedscientist.com/donate to make a contribution. That's all we've got time for in this episode. Do tune in on Tuesday, though, when Titans of Science is back, and we'll be joined by planetary scientist Sarah Russell, who will be with us to explain what it's been like to get her hands on material dating from the origins of our solar system and where we all came from. And speaking of where things came from, The Naked Scientist comes to you from the University of Cambridge's Institute of Continuing Education, and we're supported by Rolls-Royce. I'm Chris Smith. Thank you for listening, and from all of us here at the Naked Scientist team, until next time, goodbye.